Hey everybody, this is a video to explain the idea of the operon. This one's a particular operon called the lactose operon, or more specifically called the lac operon. Eukaryotic cells like you and I do not have operons, just bacteria. And basically an operon is pretty much defined as a gene or a series of genes that can control, in other words, turn on or off other genes or other mechanisms in the cell. So let's talk about the idea of food. Um, Bacteria, most of them can use lactose for food, and lactose in its form, they can't use it for food because it's glucose and galactose hooked together. But what they can do is they can end up separating them. So if I just kind of like real quick show you, here is, and this is a terrible drawing, but this is glucose and this is galactose. And together, hooked up like that, that's lactose. So that's what the cell needs, but once it gets it in, it basically has to cut it in half. That's really what it needs to do, is it needs to do this and separate these two. And then once it separates these two, then it can basically use this, the glucose. And as you learned in my previous videos, it's going to use that to do cellular respiration. So it can use lactose. And the idea here is that how I broke it apart with that blue line, I'm going to call it the scissors. It makes no sense for the bacteria to be producing these scissors to cut up lactose when lactose is not around. So the idea here is that this particular operon is going to kind of control how the cell prepares for and uses lactose when it's around and when it's not around. So these things right here are basically RNA polymerase. And let me start adding the genes. So this first one, well, let me go out of the draw and zoom here. So now that I'm out, we can see these little things moving around. That's RNA polymerase, and they're going to turn DNA into RNA and then eventually to protein. So this is part of the chromosome. If you can imagine this chromosome being one big circle, this is just part of it. The operon would start from here to here. And so basically what there is there are a couple sets of genes, and this is called the LAC-I gene. And you'll note that these RNA polymerase don't even know that this is here yet because I have to add a promoter. A promoter is almost like on a runway. It's the lights that an airplane uses to land. It knows where the runway is based on the lights. And so once I turn that on, you'll note that now the RNA polymerase knows this is where it's going to land, and it's going to turn this DNA, it's going to do transcription into RNA, and it's not showing you a ribosome, but a ribosome is going to then turn that into this. And that thing is a repressor protein. And so what I'm now going to do is add the gene over here, and I'm also going to add this thing called the LAC operator. You'll note that what happens here, boop, that little repressor sits on here. So what you're going to see now is as soon as I turn on this promoter upstream, that's going to attract these RNA polymerases. But watch what's going to happen. As soon as I put this here, here comes the RNA polymerase. Uh-oh. It can't get to this particular gene. And this particular gene, the LAC-Z, produces the scissors that end up cutting up the lactose to free the glucose that's inside of it. So this makes sense right now that there is no lactose around. So why would the cell waste its time producing all these molecular scissors to cut up lactose if there's no lactose around? So what we're going to then basically do now is I'm going to just be quiet for about 20 or 30 seconds, which I know you'll find unbelievable, and I'm going to add lactose by pumping this, and we're going to see if you can figure out what happened. You can stop the video if you want to rewind it, and then I will talk about it once the situation is over. So here comes a bunch of lactose. And so now what you're going to see is we're kind of back to where we started. And where we started now was basically that this gene does not have an operator, which basically means it's on all the time. And there's the repressor proteins, and these repressor proteins are sitting in front of this gene. So this is the scissors it's going to make. And so now I'll do it animated. So if I add a couple pieces of lactose, in other words, the cell is landing on lactose, the lactose binds to the repressor protein and pulls it off the operator. Then the RNA polymerase can go ahead and make the scissors that are then going to cut up the lactose and free the glucose from the lactose, as you'll see once it unbinds to this over time. There it goes. It's going to cut it up. Boom. Now that there's no more lactose around, look, the repressor protein is free to land again to the operator and block transcription so that this can't get here. So the whole purpose of how the lac operon works is that the presence of lactose itself, the lactose binds to the repressor protein, pulls it off of the DNA. RNA polymerases are now free to produce molecular scissors called beta-galactosidase. You don't need to know the name of that. And now look what happens. The scissors are cutting up the lactose and freeing the little golden spheres, which are the glucose, for the cell to then do cellular respiration. Once the cell's used up all the glucose, then as you can see what happens, or the lactose, there's no more lactose to bind to the repressor protein, so the repressor protein goes back and lands on the operator, so now this thing can't make any more repressor or any more scissors to cut up lactose. So the whole system basically is that in the presence of lactose, lactose binds to the repressor protein, removes it, so RNA polymerase can make copies of molecular scissors to cut up the lactose that's there to get the glucose out. And I hope you enjoyed it.